Good morning. My name is Mapori Madiehe, and with this poster, I will be discussing the environmental factors that contribute to the maintenance of Cryptococcus neoformans pathogenesis. Cryptococcus neoformans is a basidiomycetes yeast that is commonly found in soil, water, and trees contaminated with bird guano. In these environments, it needs to compete for food and space with other organisms. It also faces the constant threat of predation from holozoic organisms as well as various shifts in environmental conditions. In order to overcome these challenges, Neoformans has had to acquire traits enhancing its survival and therefore avoid extinction. However, in the course of ensuring its survival, it has become a successful human pathogen, where the gradual increase in population presenting with HIV infections and the use of immunosuppressive therapies has led to a concomitant rise in the isolation of Neoformans in clinical settings. To adequately respond to the stresses it faces, Neoformans initiates several signaling processes that result in phenotypic and physiological adaptations. These adaptations employ the high osmolarity glycerol response pathway consisting of MAP kinase, HOG1, and subsequently MAP kinase kinase and MAP kinase kinase kinase. This pathway regulates capsule, melanin, and ergosterol biosynthesis, which defends against predation, hydrolytic enzymes, and temperature shifts. In addition to the HOG response pathway, Neoformans engages the cyclic AMP, pKa, and RAS signaling pathways in order to adapt to osmotic pressure and environmental oxidants. The RIM101 pKa dependent signaling pathways are responsible for responding to nutrient limitation and pH change. Mammalian phagocytic cells are characterized by nutrient-poor and highly oxidative conditions. However, because of these pathways, Neoformans is able to overcome these challenges during infection in susceptible hosts. Therefore, we can conclude that environmental pressures have aided the evolution of Cryptococcus neoformans. Having extensive knowledge of these traits may allow for more effective control measures of these organisms in the future. Finally, I would like to thank the University of the Free State, the NRF, as well as the South African Society of Microbiology.